corona. So the factors which are affecting the rate of enzymatic activity, in that we have seen the factors. The first one is a pH, the second one is temperature, and the third one is substrate concentration. So in last class, we have seen the effect of substrate concentration with rate of enzymatic activity. And the graph is plotted here. And here, this is a graph. The rate of enzymatic activity increases with the increase in substrate concentration. However, add more amount of the substrate. However, if you add more amount of the substrate, the rate of reaction gradually decreases. So here, this is what we are calling as the substrate concentration, right? So this is the substrate concentration. This is in terms of this. In terms of this, millimoles or moles, millimoles or moles. Substrate concentration in terms of this, millimoles or moles is the substrate concentration. Now this is a Vmax. This is the maximum rate of reaction. This is what we're calling as Vmax, and this is the rate of reaction. Rate of reaction, or the amount of product formed per unit time, maximum rate of reaction, and this is in term represented with Vmax, maximum velocity of this enzyme catalysis. Now, half of the Vmax is what we're calling as Vmax by 2. This is what we're calling as Vmax by 2. Half of the Vmax is being called Vmax by 2. And this Vmax by 2 and the graph, when it has been the plot on this x-axis, this plot on x-axis, this is x-axis, and this is y-axis, and this intercept on x-axis, this gives the value of this, what here, this gives the value of this Km. Km has been called as Michaelis-Menten constant. It is what we're calling as Michaelis-Menten constant. So here, a velocity of this enzyme catalysis could be written as V is equal to Vmax into S by Km plus S. This is velocity of this enzyme catalyzer reaction. The velocity of this enzyme catalyzer reaction, the Michaelis-Menten equation, which is, this is being called Michaelis-Menten equation for velocity of enzyme catalysis. Enzyme catalysis, the velocity of the reaction, the velocity of the, now you have seen V square minus U square is equal to AS, V plus U is equal to AT, S is equal to UT plus half AT square, like that. The velocity of that, in the same manner, the velocity of this enzyme catalyzer reactions with enzyme substrate reactions, the Michaelis-Menten has given an equation for this enzyme catalyzer reactions. What is that? V is equal to Vmax into S by Km plus S. In this, V is a velocity. You may consider this is a velocity of the reaction. Vmax is a maximum velocity. S is a substrate concentration. Km has been called as Michaelis-Menten constant. And again, S is a substrate concentration. With the help of this, the velocity of this enzyme catalyzer reaction can be calculated. What is the formula? V is equal to Vmax into S by Km plus S. Now, what is the Km? This Km is Michaelis-Menten constant. This is Michaelis-Menten constant, Km. This value of the Km, this is the value of the Km. How to calculate the value of the Km? The value of the Km could be calculated by knowing the value of this Vmax by 2, and that Vmax by 2, plotting the graph, the intercept, and the plotting the graph on x-axis, then you can find the value of this Km. So a Km is the value which is equal to Km is equal to V max by 2. So Km is equal to V max by 2. Km 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 is equal to substrate concentration. Km is equal to substrate concentration at that velocity of enzyme catalyzer reaction is equal to half maxima. So right here, Km is equal to substrate concentration. Km is equal to substrate concentration at that point. At that point, velocity of this enzyme catalyzer reaction is equal to half maxima, which is equal to half maxima. Right, listen here. The rate of reaction is increases with increase in substrate concentration. However, if you add more amount of the substrate concentration, the rate of reaction, it may increases, but it remains constant, or it may decreases. The reason is that as more amount of the substrates are there, but the enzyme concentration is remains constant. So because of the more amount of the substrates are there, the rate of catalysis will gradually be decreases. In the initial phase, the rate of reaction is increases, but later on the reaction rate is gradually decreases. If you consider the maximum rate at which the rate of reaction is maximum, this is what we're calling as Vmax. Vmax is the maximum rate of this enzyme catalysis. So this is a Vmax of the substrate concentration. This is a Vmax of the substrate concentration at this point of the substrate. This concentration of the substrate where the enzyme substrate reactions are maximum. Enzyme substrate reactions are maximum and more amount of the product are being formed per unit time. And this is what we are taken consider as what here it is a Vmax. Half of the Vmax is what we are calling as Vmax by 2. Half of the Vmax is being called Vmax by 2. Now how to calculate this Km value? Km is a point of intercept on x-axis by plotting a graph, here, and half of the Vmax on the point of intercept on x-axis is what we're calling as Km. So here the value of the Km is equal to substrate concentration. The value of the Km is equal to substrate concentration. At that point, the velocity of enzyme catalyzer reaction is equal to half maxima. At that point, the velocity of this enzyme catalyzer reaction is equal to half maxima. Right, you got it? So we know that V is equal to Vmax into S by Km plus S. Now Vmax by 2 is equal to Km. Vmax by 2 is equal to Km. Km is equal to nothing but substrate concentration. Km is nothing but substrate concentration. At that point, the velocity of enzyme catalyzer reaction is equal to half maxima. That is very important. Please make a note here. Please make a note here. Vmax by 2 is equal to Km. So what is the Km? Km is nothing but the substrate concentration at which the velocity of this enzyme catalyzer reaction is equal to half maxima. By plotting an intercept on x-axis, then you can find the value of this, the value of the Km. So by this, you can find out the value of this, what here? The Km value. Right? Now, Km will provide an affinity of an enzyme towards the substrate. Km provides affinity of an enzyme. Km provides affinity. Km provides affinity of an enzyme. Affinity of an enzyme towards the substrate. Km provides an affinity of an enzyme towards the substrate. Now Km with affinity, it has got inverse relationship. Km with affinity, it has got inverse relationship here. Km with affinity of an enzyme. Affinity of an enzyme. Affinity of an enzyme towards the substrate. Km has got inverse relationship with affinity of an enzyme toward the substrate. Higher the Km, lower the affinity. Lower the Km, higher the affinity of enzyme toward the substrate. Now, for example, if this is a graph, now here we have this 1, and this is a Km value, and here there is a one more reaction. Now this is the another thing here. Now, and this is the Vmax by 2 here, and this is a Km. So this is 1 Km value here, and this is Vmax by 2, and this is this Vmax by 2, and this is Vmax. Right? So this is the substrate concentration. Now here, observe here, this is lower the Km value, nothing but lower the Km value, higher will be the affinity. Lower the Km value, higher will be the affinity of an enzyme toward the substrate. Lower the Km, higher the affinity of an enzyme towards the substrate. Higher the Km, lower will be the affinity of an enzyme towards the substrate. So if you are obtaining these values in a graph, for example, this is a Km, you can say this is a Km, and this is another Km value. Now, among these two values of this Km, which one has got more affinity means this has got more affinity and this has got less affinity. So A has got more affinity because its Km value is less, whereas B has got less affinity because its Km value is more. So Km will in terms express the affinity of an enzyme towards the substrate. Higher the Km, lower the affinity of an enzyme towards the substrate complex. Lower the Km, higher the affinity. Right? Please make a note here. This equation and this copy this. A very important for examination point of view. Please copy this. Right? Then completed. Any doubts now, please? Any doubts? Any doubts? Shweta, Shreya, Meda, Anna, Manaswini, Teja, Aditya. Any doubts? Geetika Reddy. Right. So what is a Km? Km is equal to substrate concentration. So listen. Km is equal to substrate concentration. It is nothing but substrate at which the velocity of enzyme catalyzer reaction is equal to half maxima. That's it. So here Vmax by 2 is equal to Km. Michaelis-Menten equation for the velocity of enzyme catalysis is V is equal to Vmax into S by Km plus S. Now Km will in terms express the relationship with affinity of an enzyme with its substrate. Higher the Km, lower its affinity. Lower the Km, higher will be its affinity. The value of the Km indirectly defines the affinity of an enzyme towards the substrate. That's it. Right. Next topic is enzyme inhibition. Next is the enzyme inhibition or enzyme inhibitors. What is an inhibition or inhibitors? Inhibitor. An inhibitor is nothing but a substance. An inhibitor is like a substrate molecule, but in terms, inhibits the enzymatic reaction here. It inhibits the enzyme conversion process. It inhibits or it prevents the formation of product. It prevents the formation of product. So here, inhibitors are like classified into competitive inhibitors, competitive inhibitors, non-competitive inhibitors, non-competitive inhibitors, and the other is being called as allosteric, allosteric, allosteric inhibitors, or you can say allosteric inhibitors or modulators, allosteric modulators. So inhibitors or the chemicals are nothing but the substrate molecules which are inhibits the rate of enzymatic reaction here. They are going to inhibit the reaction. They are going to inhibit or suppress the reaction. So let's start here. A competitive inhibitor. So what are inhibitors? Now in general, forget what is a substrate. A substrate is nothing but a chemical substance where it is going to bind with enzyme active site and the substrate is being converted into product in the case of normal enzyme substrate reactions. But what is an inhibitor? An inhibitor is a substance or a chemical which is going to inhibit the rate of enzyme reactions here. It inhibits the enzyme catalysis process here. So here the inhibitors are competitive inhibitors, non-competitive inhibitors, allosteric inhibitors or allosteric modulators. Right? Now let's start here. The first one is competitive inhibitor, non-competitive inhibitor, allosteric inhibitors or allosteric modulators are the three types of this inhibitors are there which inhibit the enzymatic activity. So what is the significance of this enzyme inhibition? Now this enzyme like now some of the drugs like we are taking here, these drugs they are directly inactivating some of the enzymes. They are directly inactivating some of the enzymes so that the metabolic pathway is somewhat going to be stopped in our body. For example, in the cancer treatment, the cells are undergoes for a continuous cell division, and here in a cell division called mitosis, many more number of the cells are going to form. So here the drugs are given in such a manner. These drugs they are going to act the specific here, specifically the enzymes or the protein molecules which are in turn activates this. So they are going to inactivate those enzymes which have been responsible for that. So here the inhibitors or the drugs or the chemicals, they are inhibit the metabolism. So the particular pathway is going to be inhibited in a cell. So we are in turn arresting the metabolism by giving in terms of this inhibitor called as drug. So it is one of the best treatment in the case of this cancer. And also remove the bacterial pathogens. This is the method here. And that is the significance of this inhibitors. So let's start here. Competitive inhibition. Competitive inhibitors. Now here in a competitive inhibition. Now before competitive inhibition, in a normal reaction, an enzyme combines with substrate and together will form enzyme substrate complex. Right? Now where this substrate is going to bind? The substrate is going to bind with active site of the enzyme. Right? And later it will form enzyme product complex. And later it will form free enzyme plus product. This is a normal enzyme substrate reactions which are going here. In a case of this, a competitive inhibitor, the inhibitor, now let us take here, this is an enzyme, and this is enzyme, and this part is what we're calling as active site. This part is of this enzyme is called active site, and here this is a substrate. This is a substrate here. And this substrate is going to bind with this way to the active site of the enzyme. It is going to bind with active site of the enzyme, and after the binding with active site of the enzyme, with enzyme substrate, together they will form a firm complex, like we have discussed in the case of lock and key theory, induced fit theory. And induced fit theory, the enzyme molecule is like a flexible. It undergoes for a conformational change according to the structure of the substrate and turns the substrate into product. That we have seen in the last class, like in lock and key theory and induced fit theory. So here, whenever it binds the substrate and it is giving rise to product here. But in the case of competitive inhibition or the competitive inhibitors, the substrate is having the structural similarity with inhibitor. Remember, the inhibitor is having the structural similarity with the substrate. Inhibitor and the substrate molecules both are looks like one and same. Both are having the structural similarities or one and same. They are morphologically, in terms of their structure, is same. In terms of their structure, both substrate molecule and the inhibitor molecule both looks like same. Their resemblances with one with another. Structurally they are similar. So as a result of this, instead of this substrate molecule, the inhibitor is going to bind with active site of the enzyme. Because of that, what is the reason here? Both are having the structural similarity, the structural resemblances. And because of that reason, the inhibitor in its excess concentration, it is going to bind with this enzyme active site. And as a result of this, now what happens here? This enzyme, it combines with inhibitor and together will form enzyme inhibitor complex. Together will form enzyme inhibitor complex. The inhibitor is going to bind with active site of the enzyme. And as a result of this, no catalysis takes place here. EI complex, this has been called EI complex, and here no catalysis. Here no catalysis takes place here. So what is the meaning of this competitive inhibition or a competitive inhibitor? Means the inhibitor has got structural similarity with the substrate molecule. The inhibitor has got structural similarity with inhibitor molecule. Both are structurally similar. Both substrate and inhibitor molecules, they exactly resembles to one and each other. They are similar in terms of their structure. And this substrate molecule is going to bind with active site of the enzyme. If substrate molecule is going to bind with active site of the enzyme, it is going to form the product here. But if inhibitor is going to bind, then the inhibitor is going to occupy the active site of the enzyme so that the substrate unable to join with active site. So together they will form enzyme inhibitor complex, and as a result of this here, no catalysis takes place. So ultimately by this, what is happening here? The inhibition of the enzyme activity is going on. That inhibition of this enzymatic activity or the enzyme has been inhibited here. And this is what we're calling as competitive inhibition. Right? Now, how to define competitive inhibition here? A competitive inhibition could be defined in such a manner in that the inhibitor and the substrate molecules both are having the structural resemblances are one and same. The structure of the molecule of this substrate, the structure of this molecule of this inhibitor, both are structurally, they are similar. They are structurally similar. Now, in general, if a substrate is bind with enzyme, it will form like enzyme, and here it will give rise to product. But if an inhibitor is going to bind here, This inhibitor and enzyme together will form EI complex, and ultimately no catalysis, no product formation takes place here. And this is like a competitive inhibition. Done? Completed? Yes, sir. An enzyme called succinic dehydrogenase. Succinic dehydrogenase is an enzyme. This is an enzyme, succinic dehydrogenase. This succinic dehydrogenase enzyme, which converts the succinic acid. This is an enzyme, succinic dehydrogenase. Succinic dehydrogenase is an enzyme. This converts the succinic acid, it acts like a substrate. And it converts the succinic acid into fumaric acid. Fumaric acid. So here, the succinic acid is a substrate. This is an enzyme, and it is been fumaric acid is a product of this reaction. This is a product of this reaction. Now here, the succinic acid, now, yes, this is the substrate here. It combines with enzyme, and together it will form what here? ES complex, EP complex, gives rise to enzyme plus product. This is like a normal enzyme catalysis where a substrate is being converted into product. What is the substrate here? Succinic acid has been converted into fumaric acid. Now here, this enzyme, its activity is made inactive here. Its activity is made inactive in the presence of an inhibitor called malanoic acid. An inhibitor called as inhibitor. It has been called malanoic acid. Malanoic inhibitor called malanoic acid. This inhibitor called malanoic acid. This inhibitor is having a structural similarity. It is having a structural similarity. It is having the structural similarity to exactly to the structure of the substrate molecule. So this inhibitor, it is having the structural similarity. Structural similarity. What is the inhibitor? Malanoic acid. MALONIC. Malanoic acid. This is a chemical or a substance or a drug here. This inhibitor is exactly resembles the substrate molecules in terms of its structure. So here, this inhibitor I, now it combines with enzyme E, and together will form what here? EI complex. So ultimately what happens here? No catalysis. No catalysis. But actually what happens if the substrate is succinic acid? It is converted into fumaric acid. Now here, where is the competition is now between? The competition is there between substrate, what is that? In between succinic acid, in between this malanoic acid. Both of them are acts like a competitively. They act like a competitive manner. They compete to bind with enzyme active site. In the competition, if the concentration of the inhibitor is more, obviously that inhibitor is going to bind with active site of the enzyme. If the concentration of the inhibitor is less when compared to the substrate concentration, then obviously the substrate is going to bind to the enzyme active site. So that's the reason here. If the inhibitor concentration is more, obviously it is going to bind with active site of the enzyme. If the inhibitor concentration is less, obviously the substrate is going to bind with this enzyme active site and the catalysis will going to form. Catalysis will go on. So here inhibitor and substrate molecules both are having structural similarity, and that substrate molecule is going to bind with active site of the enzyme. But in the case of competitive inhibition, if the concentration of the inhibitor is more, since the inhibitor is having exactly to the similarity to the structure of the substrate molecule, that inhibitor is going to bind with active sites of the enzyme. And as a result of this, together they will form EI complex, and ultimately there is no catalysis takes place here. Right? So here, the substrate molecules, succinic acid, malanoic acid, they are both are substrate molecules here. Both are substrates, and both of them are competitively act on enzyme active site. What is enzyme here? Succinic dehydrogenase. So if inhibitor is going to bind this active site, ultimately no catalysis. The substrate is going to bind, it is going to form the fumaric acid here. But however, if the concentration of the substrate is being increased, then this inhibition is reversed. If the concentration of the substrate is being increased, the inhibition is reversed. Please copy this. I will explain that. Copy this. This is succinic acid. The succinic dehydrogenase is an enzyme here, which converts into fumaric acid is a product. Now this is a catalysis of this equation. If inhibitor malanoic acid is going to bind, this inhibitor combines with enzyme, together will form enzyme inhibitor complex, and ultimately no catalysis will take place here. So both substrate and inhibitor both are resembles one and same in terms of their structure. But however, the thing is that if the concentration of the inhibitor is more, obviously it will occupy with active sites of the enzyme. In that competition, the inhibitor wins. If the concentration of the substrate is more, obviously the substrate is going to win that. Understood? Ardhamainda? Everyone? So everyone got it? Shushmita and others, everyone got it?
Yes, sir.
Now let us take a test tube here. Now in the substrates and inhibitors are placed here. Substrates and inhibitors. And inhibitors are less here, substrates are more. The substrates are more, inhibitors are less, and we have added enzyme. So what is the condition here? The substrates are more, inhibitors are less. Now in another test tube here, the inhibitors are more and the substrates are less. And again, the enzyme is added here. Now this is the A and this is the B. Now here, inhibitors are more, substrates are less. Then what happens here in the test tube A, in the test tube B, what happens here? Since the substrate concentration is more, inhibitor concentration is less. Here, what happens here? The enzyme catalysis. Takes place. Enzyme catalysis takes place here, but here no catalysis. Here no catalysis. Why? Because of the inhibitor concentration is more. Now to this test tube B, now here I am adding more amount of substrates here. More amount of the substrates, not inhibitor. I'm adding more amount of the substrates to this inhibitor test tube B. I'm adding more amount of the substrates. Then what happens here? What happens? In this competition, as the more number of the substrate molecules are there, those substrate molecules can replace the inhibitor at that active site, and these substrate molecules can join on the region of this active site, and the catalysis will take place. If you increase the concentration of the substrate, obviously, as the substrate concentration is more, and that would replaces the inhibitors from active site, and ultimately the catalysis takes place here. When if the substrate concentration, providing the substrate concentration is more, but however, if the substrate concentration is less, inhibitor concentration is more, obviously no catalysis. Got it? Please take this. Here, the substrate concentration is more, inhibitor or less, and here no catalysis. Here inhibitors are more, substrate concentration is less. This leads to catalysis. Inhibitors are more, substrates are less. Here no catalysis. However, catalysis could be possible. Enzymatic conversion could be possible when if you add more amount of substrate, more amount of the substrate to this test tube, then that will replaces the inhibitors, and then enzyme is going to bind with this substrate, and the substrate is going to bind with enzyme active site. So it is like a competition. If one is more with respect to its concentration, it will wins. That's it. So here you write here substrate is more, inhibitors are less in this test tube A. Enzyme catalysis, it is takes place. Here, inhibitors are more, substrates are less, no catalysis takes place in this. The concentration of the enzyme remains constant. Enzyme remains constant here and there. Enzyme is constant. No increase in concentration of the enzymes here. Got it? Everyone? Clear? Shall I erase it? Meda. Meda. Now, if you plot a graph of this, this is the Vmax value, and this is like what here? This is enzyme, substrate, ES complex, EP complex, and together it will give rise to enzyme plus product. This is enzyme substrate reaction, right? So this is the Vmax by 2. This point of intercept, this is value is what we're calling as, this is Km. This is the substrate concentration. And here the rate of reaction. Now this is for enzyme substrate. Now again there is an enzyme inhibitor also is there, and that enzyme inhibitor now so this is what your Km, and this is for inhibitor. Now here this is a Vmax value here. This is inhibitor. Now observe here in this graph the Km value for substrate, it is low, whereas the Km value for inhibitor increases. So here in a competitive inhibition, and this is for inhibitor, the dots which have been shown here, this is for inhibitor. Inhibitor means here enzyme plus I together will form EI complex, and here no EI complex, no catalysis will take place here. And here the Km value is increases in the case of competitive inhibition. Observe here, the value of the Km here, this is a Km for this inhibitor, and this is a Km for this substrate value. So the value of the Km increases, the Vmax value will remain constant. Remember, in a competitive inhibition, value of Km increases, Vmax value constant. Please take the graph and make a notes of this. This is extra syllabus, but this is there for NEET examination also. The video is not clear. The video is not clear for everyone.
Sir, it's clear, sir.
Someone is saying not clear. Shreya, Aditya.
Sir, it's little bit blurred, sir, and it's coming bandwidth is low.
It is showing that bandwidth is low.
Clear ga unda leda?
No, sir. It's blur.
Sir, clear only, sir.
Someone is saying blur, blur.
Clear.
Sushmita, how is it now?
A bit blurred, sir, and it is still showing that network bandwidth is low.
No, network bandwidth is low, but how's the board? At least, can you see this? No. Yes.
No, sir.
No, sir.
I will make it off and on. Please join again. I will make it off and on. Please join again.
Sir, both